Okay, so I think that the number of participants has stabilized, and we can start. So welcome everyone to today's episode of the International Virtual EPR Seminar. And today our speaker is Angeliki Giannouli. She's connecting to us from sunny Crete, or not so sunny today, as she told. I think in the EPR community, Angeliki doesn't need an introduction because everybody knows her. Still, a bit of a debrief. Angeliki received her PhD from the University of St. Andrews in Scotland for the work on pulse dipolar EPR in the group of Bella Bode. And afterwards, she had a long, fruitful period at the Weizmann Institute of Science, where she joined the group of Daniela Goldfarb for a postdoc to study high-field EPR. And then she moved on to the group of Lucio Friedman, also at Weizmann, where she served as a staff scientist and was researching DNP in different system dissolution DNP, heterogeneous systems. And starting last year, Angeliki opened her own lab in the University of Crete in Greece. She received lots of different awards and scholarships. I would only mention the Albert Overhauser Award of the German Chemical Society and an outstanding postdoctoral research award from the Weizmann. She stayed as visiting fellow in different places in Germany, in Marseille, in the Mag Lab in Tallahassee, and she's very much engaged in the community. She has organized and co-organized lots of meetings and conferences, and she's doing brilliant science, and we're looking very, very much forward to your talk. So Angeliki, the mic is yours.
Thank you very much, Alexei. This is very moving, what you said. And thank you for having me here today. Actually, it's been a long time since I spoke to an EPR-based audience, so I'm very happy to be giving a talk here with you today. So my talk will be on Hsp90 with the title Beyond a Passive Tether, Structural Insights into the Disordered Tail of Hsp90. This is a whole new story which is not published yet, so I hope it's exciting for you, and if you have questions, you can also interrupt me during the talk. Why I cannot change it? Just a second. I think the connection is very slow. Okay, to start with, heat shock protein of 90 kilodalton, or else Hsp90, is an important regulator of the cells, and it exists in all organisms from human, yeast, and bacteria. And it's very important because it's known to interact with a plethora of other proteins, actually more than 200 other proteins, called clients, facilitating their proper folding, maturation, and degradation, and also facilitating their the proper 3D structure. And a lot of these clients are oncogenic. So it has been proposed that by inhibiting Hsp90, we can actually target cancer. Additionally, Hsp90 is involved in processes such as neurodegeneration and therefore is a drug target also for Alzheimer's disease. In order to perform its function, Hsp90 uses energy from ATP together with magnesium ion as an essential cofactor of the ATP hydrolysis. And during its function, it undergoes small and large conformational changes which are related to the ATP hydrolysis and binding. And these conformational changes are further regulated by other proteins called co-chaperones. And these co-chaperones are known to bind either at the N-terminal domains of the protein up here or on the C-terminal domains. In terms of structure, Hsp90 is a homodimer protein, as you saw before, with the N-terminal domain up here, which is it has the ATPase binding site. So we have two ATPase sites in Hsp90. We have the middle domain, which is the binding site of the clients. And also we have the C-terminal domain, which is the site where Hsp90 is constitutively dimerized. And also it serves as an anchoring site for some other co-chaperones, which are called TPR co-chaperones and are important for Hsp90 functions, as we will talk later. You will also appreciate the structural viability of Hsp90, and I give here an example of the yeast homologue and the bacterial homologue from E. coli, and you will see that the closed structures of Hsp90 in the different forms are a bit different. So it's not, it depends on the homology, of course, but there is a lot to discover for its homologue and for its different state. And also you will appreciate how different is the open conformation of the bacterial homologue compared to the closed state, where you will see it adopts an open V-shaped conformation. So today we're going to be focusing on the yeast homologue of Hsp90, performing electron paramagnetic resonance studies. Of course, in order for someone to do EPR, we need to have something paramagnetic into our system. So we can either exploit intrinsic paramagnetic metal centers, or we can introduce site-specifically spin labels in the form of nitroxide, trityl radicals, or copper and gadolinium chelates. And here are some examples. And once we introduce them site-specifically to our system, if we have one spin label, we can look at the CWPR spectrum and derive information about the local motion of the protein at the labeling site. Or we can introduce two spin labels in our system and really measure interspin distances, this time at cold temperature, not at room temperature, by measuring dipolar oscillations using sequences like the DEER spectroscopy. Apart from the distance information, the DEER experiment also gives us information about the number of spins involved, which is now routinely used for probing the dimerization constants and also finding the oligomerization degree of homodimeric ion channels. Now moving on to our system, Hsp90 is a very intriguing molecule which has been studied extensively with other techniques but not EPR so far. So we started our studies using the standard spin labeling approach, using nitroxide spin labeling, which we introduced into different places into the protein at three different domains. And of course, we have one mutation at a time to look at. And also because the protein is dimeric above nanomolar concentrations, we directly have for each spin label protein one distance constraints. And here are the CW-EPR spectra of these mutants in the apostate, which means in the absence of nucleotide, or upon addition of magnesium AMP PMP, which mimics the pre-hydrolysis state, or upon addition of magnesium ADP, which mimics the post-hydrolysis state. And we see that we don't really have big differences in the different spectra. And this is probably due to the protein being very flexible. So the presence of the nucleotides did not really drive any conformational change, any drastic, let's be more accurate, conformational change into our system. And this is also manifested in the double electron-electron resonance EPR data, where we see that, for example, for the end domain mutants that I give here, we have very broad distance distributions and we don't have any unambiguous changes from the apo to the pre and post hydrolysis states. And also, in some cases, the distance we expect from the X-ray did not really match the one observed from the experiment. So, from this sort of experiments, we understood two things. First of all, that the protein is intrinsically very flexible, and also that the spin label introduces additional flexibility that messes things up in understanding its structure. So nitroxide spin labeling was not enough for our system. So then we, to gain structural resolution on the N-terminal domains, we exploited the magnesium ion, which is at the ATPase site, which we substituted with manganese, which is paramagnetic, and we really measured this time manganese to manganese distances in the different states of the protein. And this is already an old story, but I just refreshed your memory, where we were able to see two different close conformations of the protein in the pre and post hydrolysis state, one at 4.3 nanometers and the second one around 3.8 nanometers, with both distances having a lot of heterogeneity. But this time, because we know that we really look at the ATPase sites, we know that this heterogeneity really reflects the conformation of the protein and not any conformational freedom of any label, because we just don't introduce any label. And for completeness, we compared these distance distributions with the one expected from the X-ray structure of the protein, and we see that the distance here is around 3 nanometers. But we were happy at the time because sometimes there is discrepancy between X-ray and solution state data or frozen solution state data. So we're able to distinguish two different closed conformations of the chaperone with respect to the N-terminal domain. Now, in order to gain more structural resolution on the C-terminal domain of the protein, we were actually lucky because this domain we could label with a very rigid gadolinium label. And we performed these similar distance measurements for two mutants of this C-terminal domain of the D560C and the K637. And we probed distance distributions that are actually do not change from the apo to the pre and post hydrolysis states, and they are also matching the one from the X-ray structure. And I have to say here that these distances match the X-ray structure, although in this X-ray the chaperone was crystallized in the presence of another co-chaperone, which I'm going to be talking about soon. So the conformation of the C-terminal domains, we saw that it's practically the same, even when this co-chaperone that binds the N-terminal domains is absent. And then we decided to look more into the conformation of the N-terminal domains by reproducing exactly the conditions of the X-ray, meaning that we introduced into our system the co-chaperone SBA1. So we produced SBA1, and luckily this co-chaperone has a native cysteine, which we label with a nitroxide spin label, and in yellow here, the yellow cloud is the position of the spin label. And this time we measure the distance between the nitroxide spin label on SBA1 with each of the manganese ions at the ATPase sites. And as you see here, we measure distance distributions that are again very broad. So the system has a lot of flexibility, but are in overall agreement with what one would expect from the X-ray structure of the protein. So by these measurements, we confirmed really that our co-chaperone is binding to the N-terminal domains of the protein. And then once we confirmed that, we measured the manganese-manganese distance in the presence of the co-chaperone, and these are the data in light blue and light green color, where you see this time that our distance distribution changes, becomes more homogeneous, and is centered around 3 nanometers, and is different from the corresponding states in the absence of the co-chaperone. So we're able to distinguish a third close conformation of the protein with respect to the N-terminal domains in the presence of this co-chaperone that now matches really nicely the one from the X-ray. So from this set of data, we're able to really probe different conformations of the N-terminal domains in the pre-hydrolysis state, in the post-hydrolysis state, as well as in the presence of the co-chaperone SBA1, either with ATP or ADP, pre and post hydrolytic, while we saw maintenance of the dimer at the C-terminal domains at all times. And with these conformations, we term close, compact, and packed. And our findings, I think, were important because up to that time in the literature, the closed conformation of the Hsp90 was postulated to be always the one in the presence of SBA1, and the other two closed states were not observed with other techniques. We then looked into more details on the C-terminal domain dimerization of Hsp90, and specifically we measured the dimerization constant using the DIR spectroscopy. And for this, we specifically looked whether there are allosteric effects on the dimerization exerted from the other domains on the C-terminal domain dimerization. So to address this, we produced the full-length protein as before, but also we produced truncated variants where we cleaved the middle and N-terminal domains. And we did this for the two mutants that I showed you before. And we did concentration-dependent DIR measurements, which are known to allow measuring the dimerization, the dissociation constant, KD. Of course, these measurements are done at cold temperatures, reflecting the freezing temperature of our sample and also include some glycerol. So we had to have some measurements that are done at room temperature. And for this, we did microscale thermophoresis measurements, which also require the presence of a fluorophore in our system, and native mass spectrometry in collaboration with Michal Saron lab at the Weizmann Institute, which, however, does not require labeling, although it uses a more volatile solvent. So by combining the three different techniques at different solvents and conditions and temperatures, collectively, we were able to identify that indeed the M and N domains modulate moderately the KD of the C-terminal domain dimer. Specifically, the KD of the full-length protein is smaller than the KD of the C-terminal domain only. And now I'm going to be talking about the more new and interesting part of this study on Hsp90, and this evolves around the flexible tail of this co-chaperone. And you will be wondering what tail, but what is not shown in the X-ray structure here is a very intrinsically disordered region after the folded part of the C-terminal domain of about 35 amino acids here, which bears the so-called MEVD motif. And this motif is used as an anchor so that several co-chaperones called tetra-tricopeptide repeat domain co-chaperones, TPR co-chaperones, bind onto the C-terminal domains of the protein. And we will study this intrinsically disordered region in the presence of the TPR co-chaperone of Hsp90 called CPR6. And CPR6 structure is not known, so I have here an alpha-fold structure prediction. So we really looked at the conformational landscape of the protein in the absence and the presence of this co-chaperone. And now the important part is that why we want to look at it. This is because this complex, whereupon the co-chaperone is bound to the tail of Hsp90, it's a complex where it sets the Hsp90 suitable for client loading. And you will see here on the right for the human homolog, we have the loading of a client where the TRP co-chaperone is bound there. And interestingly, you see only one co-chaperone binding on Hsp90. So we want to focus exactly on this complex of HSP and the co-chaperone CPR6 with CPR spectroscopy. And again, we looked both at the full-length protein as well as the C-terminal domain only, which we labeled with either nitroxide spin labels or with gadolinium spin labels. And to look at the tail, we produced two different system mutations, the A687, which is marked here, and the A701. And because the protein is dimeric, we also have the position also on the other side, which is also labeled. When we look at the first position, the 687, on the full-length protein, we see, which is on the bottom, we see that we obtain a distance distribution between the two spin labels that is around 5.1 nanometers. And this distance doesn't change upon addition of the CPR6 or upon addition of CPR6 in the presence of magnesium AMP PMP. However, when we look at the isolated C-terminal domain only, we see that the conformational space of the protein at these labeling sites is different, giving a distance of about 5.7 nanometers, which, however, is remodeled once we add the CPR6 co-chaperone into our system. So we see that, first of all, that the conformational space of the C protein, isolated C-terminal domain and full length is different. And also, we see that CPR6 remodels this conformation. And the difference between the isolated C-terminal domain and the full-length protein could be due to interactions of the tail with the folded domains of the protein, which is either the C-terminal domain in the vicinity of the tail or the middle domain in the case of the full-length protein. And then we looked at the other mutant, the 701, both again in the full-length protein and in the isolated C-terminal domain. And we see that in the apostate, we obtain distance, basically we obtain data that is a flat line, which means that our distance is too long to be measured. It's outside their accessible distance range. And once we introduce the CPR6 co-chaperone there, we see that the isolated C-terminal domain does not change conformation, whereas for the full-length protein, we see a bending of the DR trace, which suggests that the distance is becoming slightly shorter, but still we cannot be quantitative to extract any number here. So binding of CPR6 for the iso-C mutant does not induce any structural change, while for the full-length protein, CPR6 binding causing shortening of the distance. And to have a structural basis of our data, of our results, and because there is no X-ray structure currently, neither for the tail nor for the CPR6, we did AlphaFold structural predictions, and we did them for several times, and we saw actually that once we have the iso-C protein together with the CPR6, we get very variable results. So this set of data we were not able to consider in our analysis. Also, we consider two molecules of CPR6 because we know from ITC data that two molecules of CPR6 are binding Hsp90 in contrast to the human homologue that I showed you before. And what we see here is that if we look on the right on the 701 mutant, both the isolated C-terminal domain and the full-length protein give a distance distribution that is above 9 nanometers, so it's quite long. However, once we put two CPR6 here, we see that the distance becomes shorter and around 7-8 nanometers, which qualitatively resembles of what we saw experimentally with the DR measurements. I forgot to say here that we did the AlphaFold and then we introduced with the MTSSL wizard software the spin label on our structure. That's how we did these calculations. For the 687 mutation, we see that the distance for the isolated C-terminal domain is slightly longer in agreement with our data compared to the full-length protein. And upon addition of two CPR6, we don't see any great changes here. So the AlphaFold distributions are in the range of the DR data we obtained. I reproduced the slight distance change observed experimentally for the APO versus the full-length protein, and also the shortening of the full-length protein in presence of the CPR6. And I think this is a very good example where DR measurements could be used for really looking and benchmarking AlphaFold structure predictions for intrinsically disordered regions. We also looked at site-specific dynamics of the tail. So we used the same mutations, but this time we did not employ the gadolinium label. We used nitroxide spin labels, which are very sensitive to their environment. And we looked at its spin label, at its mutant, in the absence and the presence of the co-chaperone. And if we take the spectra one by one, we will see that in the iso-C protein, the isolated C-terminal domain, our protein is very flexible at the labeling site, whereas upon addition of CPR6, we see that we have an appearance of a slow motion component in our spectrum, which is about 30%. And the same happens for the full-length protein at 687 position, although here we start from already part of the protein that is already rigid. However, when we look at the isolated 701 mutant, we see that both in the absence and presence of the CPR6, we have a very flexible protein at the labeling site. And for the full-length protein, we see that we have an increase of the slow motion component, although again, we started from a part of the spectrum that is already rigid. So overall, we saw that the spin label in both mutants, in both iso-C mutants, in the absence of CPR6 are very flexible. And also we saw that in the full length, the label populates conformations that are restricted, suggesting that there might be some transient interactions of the tail at this site with other regions of Hsp90, most likely with the folded part of the C-terminal domain and or with the MD for the full-length protein. Apart from looking at the tail, we also decided to look at the folded part of the C-terminal domain at three different mutants at position D560, which is in red, K627 in blue, and K637 in purple, in both full-length protein and the C-terminal domain only. And we see that for two of the positions, the 560 in red and the 627 in blue, we practically see no difference in the absence and the presence of the co-chaperone. So no matter if we have the co-chaperone or not, these sites are insensitive to both the tail presence and the CPR6 binding. However, when we looked at the other mutant at position K637, which is found really in the proximity of the tail, as you see here, and we measure the distance distribution, we see some differences between the full length and the isolated C-terminal domain protein. Specifically for the full length, we obtain a distance distribution matching the X-ray and also which does not change upon addition of CPR6. However, for the isolated domain, we see that we obtain a distance distribution that has heterogeneity and features two populations, one at 3.4 nanometers and 4.9 nanometers. And we see that the second population is going down, is diminished upon addition of CPR6. And on the right, you will see the relative populations, how they change with the amount of CPR6 equivalents. So we can say from this data that in the iso-C protein, the tail is unconstrained because there is no middle domain there to stabilize it, so it can interact with the C-terminal domain, promoting a more heterogeneous ensemble. However, in the full-length protein, there are tail-middle domain interactions that limit this engagement of the tail with the C-terminal domain, resulting in a more uniform conformation. And also we see that once we put the CPR6 in the iso-C domain, we stabilize the protein and we get a more compact conformation. Now, to directly test the contribution of the tail to the observed C-terminal domain heterogeneity, we created C-terminal domain truncated mutants, which means basically we produce the same protein, but we had cleaved the tail. And you will see here that both in the isolated C domain and the full-length protein, once we cleave the tail, the distance distribution changes and shifts to about 3.2 nanometers for both the variants, supporting a more homogeneous system. So from this set of data, we can say that the disordered tail really modulates the C-terminal domain conformation in both variants, in both forms of the protein, and its removal stabilizes a more compact C-terminal domain architecture. And now to take it a step further, we directly detected which are the contacts between the tail and the C-terminal domain by NMR spectroscopy. So for this, we teamed up with the lab of Michael Sattler in Munich, and they measured proton nitrogen HSQC, and specifically they measured chemical shift perturbations, and you will see that in the region of 560 to 570 and 620 residues, we have increased perturbations. And we have now pinpoint the residues that are affected by the presence of the tail, which are marked here on the top right with light blue color. So by comparing the protein without the tail and with the tail, we're able to see which residues are affected, and below are given some specific residues. So indeed, the disordered C-terminal domain tail of Hsp90 actively modulates the conformational ensemble of the folded C-terminal domain. So it does not only serve as binding site. And the question of $1 million comes whether these effects are relevant in the cellular context. And to address such questions, we're lucky because EPR can do this sort of measurements. We need to have our system in some sort of cell environment or cell mimicking environment. And typically what we're doing is we have our label protein in cell lysates. And in this case, we use both lysates of yeast cells and also of human cells that are shown here. Because there is a 60% homology between these two, between Hsp90 in yeast and HeLa cells. And of course, we measured our distance measurements in the presence of lysates. And also, we looked at the labeled protein directly in cells. And because we cannot introduce proteins into yeast cells because of their cell wall, we turned as a proxy to human cells, HeLa cells, which, however, we cannot internalize the protein with direct translocation due to the hydrophobic membrane, but we can apply an external trigger, which in this case is electroporation, and we managed to put our protein inside HeLa cells, and again, we did electron paramagnetic resonance measurements. So when we look at data in the yeast cell extract, we see that the distance distribution in vitro, which is the one in gray color, features the heterogeneity that we saw before. But as soon as we measured the protein inside 10 or 20% cell extract, we see that this second population gets diminished. Not totally, but we can say that actually it reproduces the effect that we saw with CPR6 in vitro. If you remember before that we saw the second population going down. However, for the full-length protein in 10 and 20% cell extract, we don't see any changes in the distance distribution in agreement with the data in vitro with the CPR6 before. Also, when we look at the protein in HeLa cells and in HeLa cell extracts, you see again that in gray is the data in vitro, whereas in the purple is the data in cell extracts, where we see again a smaller population, the second population is less, and the more compact conformation is enhanced this time. And in the case where the protein inside the HeLa cells, we see that this second population has completely vanished. Similarly, when we have the full-length protein, we see no structural change in our distance distribution in agreement with the in vitro data in the presence of the CPR6. So from this data, we can say that the cell extracts and the cells, the HeLa cells, contain endogenous TPR co-chaperones that are present and bind our Hsp90 yeast Hsp90 tail and reproduce the conformational effects observed before when we added the CPR6 to our C-terminal domain in vitro. And because we really want to prove that this is indeed the case and to confirm that we can follow, we can see in cells and in cell extracts binding of the endogenous TPR co-chaperones on Hsp90, we performed pull-down experiments followed by mass spectrometry and peptide mapping. And these are two different experiments where we really could see a plethora of proteins binding on the Hsp90 tail. However, the ones that are shown here in red are known TPR co-chaperones, which were found to bind our protein in the extracts. So this experiment really confirms that what we see in the cell stems from binding of endogenous TPR co-chaperones on our protein, and this effect also can be reproduced in vitro by the binding of the CPR6 into the tail of Hsp90. So somewhere here I finish and I say that we investigated with TPR spectroscopy the conformational space of the intrinsically disordered C-terminal domain tail and also its effect on the folded C-terminal domain in the absence and presence of CPR6. Also, we saw that the conformational sample in the isolated C-terminal domain and full-length protein is different due to interactions of the protein with the folded C-terminal domain and with the middle domain in the full-length protein. These differences between the two variants are abolished upon CPR6 binding, and the protein adopts the same conformation. Also in iso-C protein, the C-terminal tail also modulates the folded conformation of the C-terminal domain close to the dimerization site, giving rise to a secondary population consistent with tail C-terminal domain interactions as we saw with NMR. Removal of the tail eliminated this second population in iso-C and induced a structural change in the full-length protein, abolishing difference between the iso and full-length proteins. And also binding of CPR6 reduced the extent of this secondary population. And also these effects are preserved in yeast and HeLa cell extracts, as well as in HeLa cells, where endogenous TPR containing chaperones are present and interact with the tail of the iso-C protein. So with this, I would like to say that this work has been a tour de force from several postdocs in the lab of Daniela Goldfarb, Elena, myself, Arina, and Maria, which are shown here. I don't know if you can see my mouse, but you see Arina and Elena and also Maria on the right photo. And also Annika and Michael helped us with NMR measurements. So I have to say that this work has now just been accepted. So there is a lot of more detail there, which you can see. You can find it online soon. And with this, I would like to thank you. And I'm happy to answer any questions you might have.
Thank you, Angeliki. Any questions? So maybe I can start. So first, this area of Hsp90 research, it seems to be very specific, but I know that it's very, very broad. There are like the separate community researching only this one type of protein. There are separate meetings for it. There's a lot of research. So could you a little bit comment on the structure of the tail in different types of Hsp90? For example, you said that there were differences, yeah, right, in yeast, in E. coli, in human Hsp90. What is known about the tails there?
So the tail in, if I'm not wrong, in bacterial Hsp90 is not there because there are no co-chaperones that are known to bind the C-terminal domain. So the mechanism of function of Hsp90 in bacterial is different. But I cannot, like yeast and human are very similar. Yeah. So it's not, this feature is not really ubiquitous among the homologs.
Okay. And what usually are the clients of Hsp90? What does it fault?
So, for example, the N-terminal domain client that you saw that binds on the top, the SBA1, this is there to slow down the ATP hydrolysis of ATP because it locks the conformation of the protein so that a client can go there and bind. But different conchaperones work differently. For example, the chaperones that bind the C-terminal domain there have nothing to do with the rate of ATP hydrolysis. Maybe they modulate it, but they're not known for that. They're mostly known because they really lock the protein in a conformation also suitable for loading of the client. So other cochaperones, lock the protein into conformation or modulate ATP rates and so on. So it's not that they all do the same function. It really depends also what is the client that is involved into this mechanism. So for different clients, there is slightly difference, but I cannot tell you more details on this because I haven't worked with clients, at least not yet, on HSP90.
Okay, and can I ask one more question? So
Sure.
when you're measuring the KD, the constants,
Mhm.
so you're measuring them by Deer, so you have to freeze the sample, right? So
Mhm.
do you expect any differences, or like how big you expect the differences to be at physiological temperatures? Do you have any indication of this or any
Yeah,
experiments?
we measure them and they're different. So they're an order of magnitude different in this specific case, at least. For example, for the D560, we see a micromolar, a KD of 12 micromolar or something like that. And with the room temperature data, we see a KD of 1 or 2 micromolar. So there is some difference. Also, it depends if the effect is exothermic or endothermic, like during the freezing process. So we also know that for example, with the KD, with the deer, for one of the mutants, we couldn't see any changes. We couldn't measure the KD because it was below the detection limit of our
Mhm.
deer. So it was below 10 micromolar or so. But this we could measure with the other techniques. So, and
Like
also,
fluorescent probes, right?
sorry.
And the fluorescent probes,
They the fluorescent probes they didn't change much the KD. So the biggest effect we saw is more between the two mutants, the D560, which was actually puzzling, that is more on the outside of the protein. This gave higher KD compared to the mutant that is really on the bottom of the dimerization interface. We're expecting to see the opposite, if any, effect, but we saw that the one outside is the one affecting the KD more. And also the temperatures from, I mean, there is difference between frozen samples and room temperature samples, but also the frozen is like, as I said, the freezing temperature. So it's, I don't know, 260 K, 268, if I remember well, for our composition with the glycerin. So it's different, but not so much.
Thomas raised his hand. He has a question.
Hey, Angeliki, thank you for the talk. Hey, I have a quick question, and you might have mentioned already. So you showed a lot of like intermolecular distances and distributions. Now, I'm curious if I would just do intramolecular spin labeling and then just do a spin dilution kind of, so that I actually observe the intramolecular distance. Did you guys ever do any of that to look at the individual tail from the individual sub
This is
domains?
this is something I also wanted to do, and I actually tried it to
Yeah.
look at the C and M domain distances by doing, dilution afterwards. And I remember we had obtained some data, but then it's very difficult to, like, you still need to do the intermolecular distances to be able to derive the
Why?
inter. Because it was very overlapping, because all this, the way this would work is to have one mutant on the C domain where you have the gadolinium label, which is more rigid and gives more narrow distance distribution, and then to have on the M domain the nitroxide label. Okay. But then still, when you measure gadolinium nitroxide that I was measuring, I could still obtain very broad distance distributions. And then you still have an overlapping nitroxide signal. So then this was very tricky for us, for me, to measure, just because everything is very flexible and broad. So we never followed up on this because of this
But would-
reason.
but but wouldn't actually that be an argument to do the intramolecular, because you could get rid of some flexibility? Because in this case right now, the issue is that your tails are both of them moving like crazy, right? So you have
Yes.
one uncertainty times the other uncertainty.
I
If one can
mean,
do an anchor point and then just look at one tail itself, then you would get rid of one uncertainty.
yes, but there was a problem also with the nitroxide labeling. Like, even the nitroxide labels, the label itself also introduced broadening in our distances, we saw. Because we
Okay.
compared, for the C-terminal domain, we compared the gadolinium label mutants and the nitroxide label mutants, and there already the nitroxide labeled mutants were giving much broader distance distributions. So
Hmm.
I think if we really want to say something about, to obtain more rigid distance distributions, one should think of the Kapper, NTA labeling, and so on. Uh, I don't think with
Okay.
nitroxide labels. Yeah,
All right. Thank you. No worries.
no worries.
More questions? Anybody? I can ask a technical question. So you show, Katrine, please.
Hello, Angeliki.
Hi, Katrin.
Hello, do you hear me? There.
Yes.
Very good. Thank you for the talk, and nice to see and hear you again. Sorry, my camera doesn't work here. I was just interested, you were saying you're doing the AlphaFold modeling when you look at the interaction of the CPR6 co-chaperone with the C-terminal
Mhm.
domain there. I don't think I heard which of the alpha folds you were using, because we sometimes see that it makes a huge difference if you use the version 2 or 3.
I think this one we did with the 3.
Okay. So
I
that's what I would have
but
set my
I
argument.
need to check. But
Mm.
there This AlphaFold really troubled us, and we had to run them a lot of times.
Okay.
Because as I mentioned, when we had the isolated seed domain with the CPR6, then the CPR6 would be in open conformation, in closed conformation, basically would be all over the place. There was no consistency in the output. I mean, you still get the 5 outputs there, but
Mm-hmm.
for the others, at least it was consistent. For this
Yeah.
combination, it was not. So we
Okay.
completely dropped it. Yes.
So you repeated the 5,
Yes,
like
we
the
repeated them many times
okay, and it's still
because
not
I
right.
was going crazy. I was like, how is it possible, And
Yeah.
yeah, this troubled us a lot.
Okay. 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 Well, yeah. Thank you. Thank you.
Okay. So if we don't have any more questions, somebody? Yes, please.
Hello. It's a good talk, great talk. I have one simple question, like, when you get that complex, the Hsp90 and co-chaperone, how do you make that complex? Just to mix the 2 proteins?
So we did surface plasmon resonance measurements to estimate the KD of the binding of the 2 proteins, and we saw that it's very low. It's about 1 micromolar with or without the label. It's more or less the same. So we know that at the concentrations we use, which is above 100 micromolar, we have the complex there, and also to ensure that we have enough, we have a 4-fold excess in most of the measurements as you saw here. So we are really sure that our protein is bound there.
Oh, okay. So thanks. I got another question is like, instead of nitroxide, have you tried any TAM radicals?
Sorry?
The TAM radicals, like a triaryl methyl.
Tritals.
Trityl.
Yeah, tritals, yes.
No, we haven't tried in this protein any trityl radicals, no.
Yeah, probably, it's a kind of the trityl may not be a good candidate because trityl is slightly bigger size. That may change the complex formation of the co-chaperone and the protein. I'm not sure. Okay, I'm just thinking because trityls are slightly bigger molecules compared to nitroxide,
Right.
Yeah.
Right.
Yeah, thanks.
Thank
Go
you.
ahead. If you want to say something, go ahead.
No, I'm wondering because, for example, when we had the gadolinium label, which is bigger than the nitroxide label, and we did the labeling on the M and N domains, what we saw there is that we had cleavage of the protein in the SDS page. So I think the 2 big radicals, I think they really the protein is not happy with big, but maybe trityl is different. I don't know. We haven't tried really. Maybe it's worth trying.
Yeah. Okay. Thank you.
Thank you.
Okay, any more questions? So if we don't have more questions, maybe we can all thank Angeliki once again. Angeliki, it's a great work and it's a great talk.
Thank you for having me.
Congratulations on this paper being accepted. We will all read it.
Thank you.
So in 2 weeks from now, we're supposed to gather back in the seminar. And I figured that there was some confusion with times because the seminar time is attached to the time in the US. And now they have already changed to the daylight saving time. And the Europe still hasn't. So from next week on, I think we're back to normal schedule. So sorry to anyone who connected later and couldn't hear the whole talk because of this time change. Maybe we didn't signify it well enough. Anyway, thanks to everyone who attended, and see you next time. Thank you very much, Angeliki.
See you. Thank you.
Bye-bye.
Bye.